Doing this a long time.
Well, welcome everybody. We're going to begin, and Bob Martin has kindly agreed to referee this herd of cats. So, Bob, take it away.
Actually, I agree to give a slight technical background. I would never agree to try to referee this group of cats. Okay, I have 2 slides. Okay, so first, what is CRISPR? Cas, by the way, is a CRISPR-associated proteins. I guess they decided for that acronym rather than CRA proteins. But anyway, the easiest way to think of these guys is just a family of customizable gene editing tools. They're very analogous to text editing tools, the one we all use. We find things, we cut things out, and we paste things in. And that's really what CRISPR-Cas does. The finds alone can be very valuable. They've been used to diagnose diseases, what's happening, been used for drug screening. And if you combine those finds with another biological paste and edit tools, you can then literally edit our DNA or RNA. You can edit RNA with Cas9. You use a different RNA. And the way this works, there you see what CRISPR means. And what CRISPR means is they're chunks, just little chunks of a bacteria's DNA or RNA that has repeats, that's the R, regularly interspaced, that means they happen frequently, and they have this funny property that if you read them from the right or from the left, they look the same way. That's what a palindrome is. It consists of 2 parts. This is the one that the folks got the Nobel Prize for. Cas9, that's one of many proteins that bacteria have. It's a very big protein. So it's 4.1 thousand base pairs. Our average protein is 539 base pairs. So this is a big protein. And it does a bunch of things, as we'll see. And then something called guide RNA, which has 2 pieces. The CRISPR RNA that matches the virus, and the tracer RNA, that's the glue that holds the guide RNA to Cas9, forming the CRISPR complex. So that's what it is. Okay, what happens, hold on, let me get rid of that and move there. The way the selective find works is the CRISPR RNA is quite small. It's only about 20 base pairs. We'll get back to that in a moment. Remember, our average gene is like 530 base pairs, so 20 is just a small part of the gene. And Cas9 does the selective find. And the Cas9 role is to find something called the PAM. The protosensor associated motif is just 3 base pairs which act as an on-off switch. That PAM occurs in our genetic sequence. It does not occur in the bacterial one. And where that's important is that makes sure that this complex doesn't eat up the bacteria. So that's the role of the PAM. And why aren't you? There we go. Okay, what Cas9 does is it splits our DNA, and then it does an exact double cut. So it cuts out that roughly 20 base pair sequence. They're actually a little bit more, but that's not important. And then to do the paste, what happens is people stick in a little bit of DNA and use the inherent repair technology that our DNA has. So that's how it works. Now, it turns out it's very hard. Think of when we do text find, and we said we wanted to find the AR in barnyard. Well, which AR, the first or the second? Well, we wanted barn. Okay, so find barn. Darn, we found this word. And that happens with CRISPR-Cas9. It's called off target. So you find the wrong thing. Before Al's work, which he described in his great lecture, when we would try to find words in text, it was really very, very hard, and we missed them. That happens with CRISPR as well. If you cut the wrong letters, which happens, if you think of the Japanese term for Catholic, you could have some unintended swear words, some wrong things could happen. If you changed a single letter in shot to eliminate gun violence, you might get rid of shoot in the arm or shot in the arm, which we wouldn't want to do. So there could be unintended consequences. I might do the cut and forget the paste. The find, cut, and paste in a rapidly evolving document is a losing race. Every day, 2 trillion of our cells divide and form new DNA. Cutting after pasting could undo the change. It's called recleavage. In other words, you could paste, but Cas9 could say, hey, I see it again, let me do it again. And just like the word run has 635 English meanings, a gene's protein has many poorly understood functions. The example that we read about in the book was the HIV baby fix. It also statistically reduced the baby's life expectancy and increased their viral susceptibility. Every example I've given here happens with CRISPR. And one that came out just this week in Nature Genetics is when you do a cut, it actually might rearrange the genetic sequence, causing problems. So things are continually being discovered. It is blazingly complicated. These are some of the some of the Cas proteins. They're broken into classes and types, and each of them have different functions. Cas9 is the one we read most about because even though it's had all of the problems I've just gone through, it's the best of the bunch. Okay, where's the action? Well, the bacteria discovered this a few billion years ago. And as you can see from this timeline, it's been moving very, very quickly. From first being understood in 1987, more details in 2005, 10, and 11, then the Nobel Prize work, whose operation I just described, was written up in 2012. Isaacson's villain. Isaacson likes heroes and villains. Steve Jobs was a villain. You could spot his heroes and villains in this book. One of his villains used it for gene editing in 2013. And then there have been more and more progress with the two big ones, one we discussed last time of turning genes on and off just April of this year, and an improved pace also in April. I would love it to be able to produce naive T cells because us old folks, T cells are wearing out and be neat if it could. There is lots of interest. There's a journal that was founded in 2018. There's a gene editing news. There's special journal issues of the big journals, Cell, Nature, and Science. If you just do a Google search, you get 60 million hits. By the way, COVID had 100 times more. By the way, sex only had 2/3 of the COVID hits, so I guess more people are interested in COVID than sex, but you know, what the heck. Now, I'm not going to go through all this, but I think the astounding thing is for something that basically was discovered 8-9 years ago, it is already being extensively tested for cancer therapy, the first, for things as bizarre as eliminating malaria, mosquitoes, for better crops, better animals. That said, the field is moving very gingerly. There are only 46 CRISPR FDA clinical trials out of their 380,000. Most of her cancer and sickle cell anemia, none for COVID-19, and there are only 6 FDA gene editing drugs. So those series of problems that I mentioned are behind why people are moving so gingerly while at the same time rapidly progressing the basic science. CRISPR is very active in COVID. There are 202 papers, small percentage of the total number, and they're in drug discovery, testing, that's most of the papers, by the way. One was approved in May. Understanding the disease, its sequence. I think its most exciting news, which I expect is going to come up in the future, is for antiviral. That paper was in May in Cell. And basically what the idea was to come up with a CRISPR sequence that could recognize the virus and snip it. They very cleverly called it Pacman. I'm not going to go through that. And I am finished with my screen sharing. Let the discussion begin. Where are you? Zoom. Yeah, there we are. Okay.
You got to unmute. We didn't hear you.
That's a good point. Asking you to raise your hand if you have a question. Some people have already done so, but just to remind you, so we can have the discussion in an orderly fashion, and Joel will call people out in order. So Joel?
Rich Jigger.
Thank you, Bob. You were focusing on Cas9, and the thought occurred to me, the question occurred to me, is Cas9 a naturally occurring protein that is busy at work doing stuff, perhaps similar to what you were talking about, even aside from any human intervention? Is this going on naturally, what Cas9 is and does and can do?
All I have read is about Cas9, and by the way, there are multiple versions of Cas9, to make it even more complicated. One was discovered about 2 years ago that's a third the size of the first one, which makes it better for gene editing. All I have read about, but I'm not expert in Cas9 by any means, is its role as part of the bacteria's adaptive immune system. That doesn't say it didn't do in other things, because as I said, it's a big, complicated protein.
Thank you.
Bill Tittle?
Yeah, I'm just amazed. If I was dreaming up how CRISPR worked, I just never would have come up with this fine cut, paste. From an evolutionary process point of view, you divided this into two parts. There's actually the third one, if I can, the tracer RNA step. That's the thing that Emmanuelle Charpentier was nervous about. She didn't get it. The idea that evolutionary, maybe the first part, the finding part evolved, but if you didn't have the pasting part, that die off. Then you had to have these three things had to evolve together, or this process never would have happened.
Yeah,
isn't that miraculous? I never would have, if I was contemplating this, how evolutionary wise it happened, I never would have come up with this.
and that it happened in a bacteria billions of years ago.
Yes.
I mean, we didn't develop that defense.
Yeah, that's true. We didn't get it. They got it.
It's just,
Yeah, it's.
Remember, bacteria have nothing to do but reproduce, and they do it fast.
sounds like a fun way to spend your day.
Okay, Walt Meisner?
I take it the Cas9 is something that the bacteria makes. Is that correct?
Yeah.
Okay. The other thing is, you said that it cuts the rails of the DNA, and what's to prevent the DNA from unraveling at that point?
No, great question. Basically, what naturally will happen with the DNA is there's something called a DNA repair process, because our DNA breaks every now and then. And what it does is glue it back together. And what the typical cut will do, if you don't do the paste, is something called a gene knockout. And let's see, sorry, here we go. Here's the DNA. It gets cut. It pastes back together. Now, when it pastes back together, most of the gene is still there. Remember, our average gene is 539 base pairs, some shorter, some longer. The longest, I think, is 20,000 base pairs, just to scale it. But when it's glued back together, it's a non-functioning gene.
Mm-hmm.
So it put an error into it then.
Yeah. Now, the other thing that can happen is something might get inserted. That, a mutation might happen. and that might mess it up. But, as I went through with all those examples, based on the little reading I did on CRISPR, man, it is a statistically unpredictable process.
The other thing is, when I took this epigenetic course a while back, they talked about cell division, and the DNA just splits down. You get two rails,
Right.
and then they fill up the missing piece at either end. And apparently, for the first 24 hours, there are molecules that travel up and down the DNA, and they will fix errors.
Right.
Okay, but after that 24 hours have elapsed, then it's impossible to, the cell has no defense against errors after that.
Right.
So I thought I'd mention that. But anyway, the other question I have is, after they decoded the human DNA, they finally had the gene sequence and they could see what proteins were being made. And I was looking at this in, late 90s, early 2000s. And the big issue at that time was protein folding, because a protein could be folded several different ways. If it's folded the correct way, it's effective. It's folded any other way, it's useless. In your deep dives, have you uncovered whether they solved that? I mean, the big issue was, they could look at the DNA, the codon sequences, and they could see what proteins were being made. But the issue really is how the protein is being folded. If it's folded correctly, it's effective. If it's folded any other way, it's useless. But I haven't, in the last couple of decades, I don't know if they've made any progress in that. Have you in your deep dives found anything in that area?
Found two things that I'm going to add, one on epigenetics that I read this morning, which is fascinating. A bad folding actually causes mad cow's disease. So
Okay.
Progress has been made in understanding folding. A Google program was almost 90% correct at predicting correct protein folding. So it happens. The details of protein folding are not yet fully understood.
Yeah, I guess the real question is, how does the body, how do the cells force the protein to be folded in the right way? What do they do to make that happen?
Okay. Simplistically, it's a thermodynamic process where it's trying to get to the lowest energy position.
I see. Okay.
No. I mean, think if you start to fall, you fall down. You just got to your lowest energy position.
Mm-hmm.
So all proteins of folds are in the lowest energy position. Is that right? Okay.
Yeah. I mean that. Now the bizarre epigenetic thing, and you mentioned this particular part of the gene last time, where the gene wraps itself about, and that.
Yeah, the histone with their tails on them.
Yeah. COVID does an epigenetic change to that improves the adaptive immune system.
Yes, I think you mentioned that before.
Yeah. Yeah. I mean, it's crazy.
Yeah.
Okay, thank you.
Yeah.
John Tomaszewski?
Yeah, I have a question. When they do the insertion of this little tiny piece, I'm assuming that they put like a massive amount of these little pieces in the part that they want to insert, not just one little piece. Is that correct, or is it down to the point where they only have one and they put that one in the right place?
Okay. John, regarding the numbers, I'm not sure. I've read about the process that is now being used. And they use most recently this secondly discovered Cas9 protein, the smaller one, and they build what's called a plasmid. They put the Cas9 protein, the tracer RNA, the CRISPR RNA, and the DNA they want to put in.
Oh,
Okay, and they attach that to a virus, the same one used in the Johnson & Johnson and the AstraZeneca, and they send that into the cell. So they get that in, and then the cell starts to do its thing.
so it's kind of like a factory almost that goes in and
Yes,
first it does some work and then it grabs a piece off the shelf and jams it in there.
right.
Thank you.
Yeah. But I'll tell you, I'm hanging on by my fingernails on this one.
Al, Aho?
This is a fascinating area. Is there a theoretical limit on how many edits you can perform on a gene? So is it possible by this technology to transform a penguin into a platypus?
Well, I know they're doing multiple edits at one time on a gene. That is being done. I didn't read anything about any limits. Obviously, you could do it sequentially, take you a long time. How many you could do at a single shot? I don't have a clue.
Okay. Thank you.
Any other questions? I actually do have one for you. What do you foresee as the future for CRISPR technology?
Yeah. This is a guess by a computer scientist who happens to read a fair amount. Okay. Yeah, as compared to by somebody who knows about this stuff. I think within 10 years, it will have transformed cancer therapy. Because that's not gene editing per se. It's adapting our T cells to attack our tumors. And this was pointed out in Jennifer's wonderful book, as what she hoped would be the big breakthrough. And you could see the progress being made in that. So, I think that's just going to happen. I would guess that the gene therapy to go in and change germline genes, to get rid of Huntington's.
Right.
Let's pick the easy one. As compared to get rid of 5'9" Canadians. That's the harder one. My guess that one is 20 years off. It just, it sounds really tricky, and it has the ethical problems that I don't know whether you guys talked about.
Yeah, ethically, it's really scary.
Yeah.
Thank you. Bill?
Yeah, I wanted to follow up comparing the challenge of managing something like nuclear fission with managing this process. Because I think it's much more difficult. We have this huge system for making sure that Joe Blow doesn't make a nuclear bomb or something like that. It's easy, but this thing, they're kits, aren't there kits I can go out and buy to do gene editing? And wow. So I'm trying to get this to be I think we're doing pretty well from what you're saying. There's all this kind of activity, and no nutsy thing has happened yet.
That we know of.
That we know of. Good point. So I guess we just have to hope that we don't screw this thing up. But it's such a blessing. All these things that you're talking about. Transforming cancer therapy. God, what a blessing that is. But then the dark side.
Yeah, well, so Bill, if you're one of the Wuhan lab conspiracy theorists, you might say that, gee, I guess they use CRISPR to modify.
Yeah. Okay, thanks.
Griff?
Hi. How do we understand the matching system well enough to ensure that we aren't making unwanted changes to other parts of the genome?
No.
Because I was listening to a person complaining that CRISPR is not going to go anywhere because they find cases where so many other edits to other parts of the genome, and they're not necessarily beneficial.
Right. Yeah. Those are called off-target edits, and the base, bad play on words, problem is there are only 20 base pairs in the find. And, we have 3.2 billion base pairs. So you might hit the wrong guy. Now, I'm making up an evolution of the technology to suddenly, and I'm making this up, I haven't read anything on it, to where suddenly the matching is 1,000 base pairs or 100, but that, I made that up. But then how you do account for little mutations? I mean, it's tough. Okay, I have a question for you guys. A highly regarded company has completed phase 1 and phase 2 clinical trials on CRISPR to create naive T cells in older people that will improve their response to all infections. Would you participate in a phase 3 trial?
Define a T cell.
A T cell is that part of the immune system that is very important in how we respond to infections. And they either respond at the first level, when the infection first comes in, or they get trained to respond when it comes back in.
Thank you.
And what happens with those T cells, they are created in our thymus until we reach puberty, and then they stop being created. And that's where the T comes from, by the way, thymus. Okay.
So we can vote, yes, no to Bob's question.
So how many would participate in the trial?
Okay, you use your reactions button, use a check mark as a yes and a X for no.
Let's see, there we go.
And so far, there are 7 votes, 9 votes, 6 of them being yes and 3 no.
Okay, I have another one.
Wait a minute, wait a minute. So
The-
we're still getting votes.
The,
It's 65.
okay, the same buildup. This would suppress the genes that cause Alzheimer's. Same buildup. Okay, good phase 1, good phase 2, highly regarded company. How many would,
So far, we're all positives. 5, 6,
Yeah.
8, 10, 11, and 2 negatives.
Oh, well, it cost me $65,000.
no, it's free.
Oh, okay.
Interesting question, yes.
Yeah. By the way, there's also a related question, and that people are closing in on some of the genes that they think cause aging.
Yeah.
Same question. Silence those genes that cause aging. Would you participate in the trial?
So far, it's 5 negatives and 4 positives, 5 positives, 6 negatives, 7 negatives, 6 positive, 8 negative, 6 positive.
Okay, so you aren't worried about getting sick and you aren't worried about dying, but you're worried about going goofball.
You got it.
There's also the rather ethical issue with the T cells of if you improve the T cells, it means more people are going to live much longer. Is the economy able to handle a massive number of old people? We can barely do it now.
Right.
Yeah. Yeah.
That's difficult. Just look at what Japan is going through now.
Yes.
Yeah. And the dropping birth rates around the world, including the US.
China's allowing them to have 3 babies instead of 2 now.
Yeah.
John, you had a question before?
Yeah, from when they started going through the DNA and finding out things, they found what they called junk DNA, and that was a majority of the DNA. And from what I understand, some of the stuff that happened in CRISPR was that when they started cutting things, it was cutting stuff in the junk DNA, and they found out that, whoa, some of this stuff is important because it was causing things that shouldn't have happened. Do you know anything about that?
I had not read about CRISPR cutting the junk. The one thing that seems to be happening, the more and more we understand DNA, and by the way, we do not yet fully understand it. There's still about 3% that is not understood. But the more that's understood, the term junk has gone out of favor.
Yeah, there's some indication that the so-called junk DNA is actually used in regulatory processes.
You know, which means the turning on and the turning off.
Right.
And the whole thing needs to be a structure, 3-dimensional. And if you cut a piece of it, then you're messing with a structure, which can mess with a functionality that might be like 1,000 base pairs away, but in reality is where you need to have the magic lock mechanism for the key to come in.
Right.
That's where I read somewhere that in this junk DNA, they some switches that were turning things on and off stopped switching. Yeah. So they had to stop, slow down the study that they were doing to figure out what went wrong.
Right. I mean, it is just so exquisite, you know, and how it works and doesn't work. I mean, it's really, you know, it's magic.
I want to comment on that, Bob. And you know, we many of us have spent some part of our career in chemistry, chemical engineering, or tangential to it. And there's all sorts of catalysts. You know, every car we drive has a palladium catalyst underneath the chassis to take care of the carbon monoxide and blah, blah, blah. There's a lot of catalytic stuff out there that we use in every refinery and so forth. All those catalysts all operate at really high temperatures. And in our bodies, we got a zillion catalysts called enzymes, and they all operate at about 37 degrees C. And we've never, I don't remember ever hearing of an industrial catalyst operating at 37 degrees C. But it's amazing, you know, our human bodies have a million of them, or I don't know, whatever, but how, you know, why can't we somehow mimic? And also the catalysts we use tend to be, you know, rhodium complexes, or, you know, mercury, or some organometallic, and most of the enzymes are just plain organic. It's just fascinating that after 150 years of all the chemical research, we're still so primitive in catalysis.
Yeah.
Al, any hope?
You mentioned that some snippets have the palindromic structure. They're the same read forward and backward. Why did this happen? Is it known?
Yeah, because the bacteria discovered a probabilistic push-down Turing machine. Now, that was any computer science thing. I haven't read why, Al. I don't know.
There's also a related question I had of why is the Y chromosome getting weaker?
And where did you read it's getting weaker?
It's a pale imitation of the X chromosome.
Why does it mean it's weaker? And what did you mean by weaker?
This was an article I read a long time ago, and it said males are in trouble.
Oh, well, that's a separate topic.
Okay.
I just read something about that, Al. It seems that that study that was 20 years ago, they're now, I forget the exact thing. It was just one of these little blurbs that I read that said that, that they don't believe that that's happening as they thought before. And but that just kind of stuck in my mind when I saw that, because I remember thinking about, God, this could be bad. And it came out that recent research said that it may not be as bad as they thought.
I'm glad you're reassuring us.
Was that Mad Magazine?
It could have been.
I know your reading taste, John.
Thank you. You know, I'd like to take a little question for everybody, if I can. I understand that CRISPR can be used in a pregnancy in the baby, and some of the thoughts is that it can give what's called designer babies. I just wondered how many people would think that this would be a good thing as a bad thing. And what I mean by that, you know, of course, everybody thinks of blue hair, blonde eyed babies, but it could also mean that they could stop diseases before they spread. So what's your thoughts on that as far as a vote? Would you want this to continue, or should they not do this type of designer stuff?
If you want to vote, vote yes if you want to allow it, and no if you don't want to allow it.
John, I think you're making this a all or nothing question. I mean, there are certain kinds of designer aspects that I think might be acceptable and others not.
Right. But I'm just saying just in general, all of these things? I guess it's a hard question because, yeah, if they find out that your baby has a disease and they can cure it in vitro, I guess everybody would vote for that. Is that correct?
I
Yeah.
certainly would.
Okay,
Yeah.
so let's take that one out of it. And just say that if we can, create the perfect athlete. If your kid could be the perfect athlete, the genius, get the genius gene and all these other good things that they might be able to insert, how many people would allow them to do that?
6 no's and 7 no's and 1 yes. 9 no's, 10 no's.
That's pretty universal.
So John, here's a hypothetical, okay? We create a perfect athlete, you said, that can run the 100-meter dash in 8.2 seconds. What's the world record now? 9 or something? So they can do it in 8.2 seconds. But then all of a sudden, every country has a designer sprinter that can run 8.2 seconds. So now we've re-leveled the playing field in, at a faster rate. And this goes, of course, to accuracy of shooting basketballs and whatever else you want in terms of sports. But just simply, if the United States can create these people who will outperform the Olympic athletes, then every country can immediately follow. And so you've just changed the game from, a superb athlete running it in 9 seconds to 8.2 seconds. But, to what advantage other than simply you've tinkered with humanity?
Well, didn't the Russians
Go to the scientists, not to the athletes.
didn't the Russians do that with chemicals?
Yeah, but,
A lot of accusations.
and the real problem is how do you figure out what the line should be? So, understanding soft-spoken people is very hard. It puts them at a disadvantage, and it puts the people who's hearing them very good at a disadvantage. So would you want to fix that so that all the voices were loud enough so that everybody could hear them? And they might become a little more uniform that way. I mean, man, it's tough.
Well, I on that question, I think I would rather see something that would fix my hearing. I am a hearing aid wearer, and go at it from that direction rather than messing with the genes that might affect how loud your normal voice is. So if you got a CRISPR for hearing problems, I'm willing to be in that trial.
Okay, but then with your very good hearing, you might hear more animals that you don't like, and you might kill those animals, leading to an extinction. So is that good? I mean, man.
Yeah, well,
you can take either side of this and go crazy.
yeah, I wouldn't go that far, and I would not elect a super hearing like Mitch's 8.2 second runner. Just normal. Just get me back into near the median. That would be fine. Thank you.
I might kill some of the lawn service people around though.
Well, one of the other aspects is the agricultural aspect, and we've been tinkering with, let's say, wheat for, somebody tell me, 5,000 years, 3,000 years, something like that. And the push that Norman Borlaug did in the 1950s, that increased the capacity. Many things have gone on, and of course now pioneer hybrids and DeKalb hybrids and all those kind of things are normal in our agriculture, as well as many other artificialities. So we're by far not eating anything remotely resembling what was on this earth 5,000 years ago. Having said that, there's been a hue and cry for against GMOs for the last 10, 20 years. And now we're looking at CRISPR redesigned plants, animals, and so forth. I just see that as a logical evolution of that same process that's gone on for 3,000 years, where people use the current technology to improve crop yields, reduce vulnerability to fungus, make it taste better, all kind of stuff. And here we are in Jersey, where we're the king of flavor and fragrance labs across the country. We make all of the funky flavors that go on. So it's just, to me, CRISPR is just the next logical extension in that world, which has a completely different morality than tinkering with humans.
As viewed by some, because one of the CRISPR tinkering is to make, and literally this is underway, to make pigs better organ donors for humans.
And somehow I'm okay with that, but that's just a gut feeling, not any kind of
right. Yeah. No, I am too, but I'm just saying, you could imagine what the PETA group is thinking about that.
Sure, and we're all dancing around a term called the slippery slope, and the fact of the matter is, like in agriculture, that slippery slope started 5,000 years ago, and we're still sliding on it. And all of the medicine we've been doing, particularly in the last 100 and so many years, to lengthen life. I mean, wasn't too long ago when, if you lived to 20 years old in Manchester, England, that was about the average during the Industrial Revolution. So we do a lot of tinkering, and it's all slippery slopes. And it's just a matter of when somebody says, whoa, 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 this is too slippery a slope.
Right.
I think my slope was slippery because part of my genome reduces the urge to reproduce in my offspring. Now I'm down to one grandchild, and I think it's going to stay that way.
Bob, thank you so much again for providing terrific insights into current research. I have to tell you, as a non-scientist, after I read the Isaacson book, I was both exhilarated and terrified. And some of the research that you talked about today, if anything magnifies that.
Which part? Both?
Well, I was a little surprised, for example, when you talked about the HIV babies in terms of reducing the longevity.
Yeah.
And so I'm with you. I'm all in favor of finding a cure, hereditary cure for Huntington's disease. But I'm kind of terrified that, we may have a second coming of Mary Shelley here if we're not careful.
Yeah. Oh, absolutely. No. I think all the constraints that the biologists have been lobbying hard for on germline editing are absolutely correct. And if anything, the unintended consequence of the Chinese experiment, might have helped that.
Great.
Which Chinese experiment are you referring to, Bob?
Oh, in 2018, unknown, as best everybody knows, to the Chinese authorities, a Chinese scientist edited basically the cells of twins, took them out, put them back in to fix a gene that is associated with HIV, to make sure that those two girls could not get HIV. And there were, he thought he was the hero advancing. And there was literally a world uproar. First, it wasn't required. There were other ways to solve the problem. So he was only trying to advance the science, not help the babies. And second, it was found out that those genes had other functions, and the fiddling with them statistically resulted in a lower lifespan and increased susceptibility to viral infections. And we just don't know what a particular gene's function is every place.
And he ended up in prison.
Yeah.
Yeah.
One of the things that struck me as a non-scientist, although I was a math major, is from Isaacson's book, which I have not actually read, but I went through Bill's notes and Mitch's notes, is that it seems like the information multiplies tremendously. The number of Cas proteins just keeps multiplying. Mitch talked about the number of enzymes that are at work in the human body. And it seems like the more we know, the more we realize we don't know. And of course, that's part of the, I'll second being terrified as a result of all of that, which apparently we do not know. And so, yeah, definitely, I think we should be very careful about anything that affects humans.
Certainly germline editing, for sure.
I just like to make a comment, like in agriculture, they've gotten the varieties of, rice, wheat, and corn and all that kind of stuff, down to 7 varieties. So they're planted worldwide. And if any one of those varieties ever are susceptible to some kind of disease or blight or something, you could wipe out a significant part of the food supply of the world. So that's like a danger of having, a very narrow set of, species to rely on. If they ever do germline editing or, or selective breeding or whatever method they want to use to do that for humans, you basically also would be reducing them to a very narrow genetic line, which again could be susceptible to, some kind of disease that just wipes out the greater majority of them. So having this diversity is actually a benefit. So other than perhaps trying to get rid of unintroduced, inadvertently introduced genetic diseases, to do anything more than that would be, to the disadvantage, I would think,
As an aside to that very point, this is something that's happening with the banana that we're eating today. Essentially, there's only one species that's used worldwide, and it's undergoing some kind of a blight, and it will be wiped out within the next 10 years. So there's a mad rush to find another variety of banana.
Yeah. Actually, the first variety of banana found was called the Gross Michael, and that suffered it was in production for decades and it suffered some kind of blight, and they had to go to this other variety called Cavendish.
Right.
And it's actually not as good tasting as the Gross Michael variety. But now the Cavendish is under threat of a blight. And if that disappears, you know, we're down to eating plantains, which are really not that tasty. You know?
So you're saying that getting rid of bananas and banana skins is a slippery slope?
Probably.
Well, gentlemen, our hour is up. I'm cognizant of the time that Bob has shared with us. I'm very grateful for his leadership. But I propose that we terminate this discussion. I think anyone's welcome. It's the old guide site is open here. Anybody can stay on and continue the discussion. But formally, I'd like to suggest we terminate the meeting. So thank you, Bob.
And Joe, I'd like to thank you for taking the leadership in starting this discussion, because I think it was very worthwhile. So I appreciate it.
Thank you.
Yeah, no, likewise. And I'm going to scoot my
Yeah, you got family here.
my New York daughter who fell and her son-in-law in here, and so I'm going to say goodbye.
Okay.
As always, good to chat with you guys. Bye-bye.
Thanks,
Thank you, Bob.